Thank you very much for that kind introduction. It's my absolute pleasure to be able to be presenting to you today at the Highlights in Medicine Annual Conference on some work that I've been very engaged in over the last 18 months. It's been one of the busiest year and a half of my entire career, and I'm going to be speaking to you today in this section on leaders in research with respect to the World Health Organization guidance development during the COVID-19 pandemic, and share some personal experiences and work that I've been doing in this regard. Just firstly, to begin, a territorial acknowledgement, since I'm presenting from within Calgary in the traditional territories of Blackfoot and Treaty 7 peoples, including the Siksika, Pakunia, Kenai, Tsuut'ina, and Stoney Nakoda nations, and the Métis Nation of Alberta Region 3. Just by way of disclosures and acknowledgements, I have received funding in the past to attend a meeting on hospital-acquired infections from the CDC and from Baramayu, who helped to fund this meeting. I hold grants from multiple peer-reviewed agencies of note from a pharmaceutical company for a randomized controlled trial of a Staph aureus vaccine. But all the funding came to the University of Calgary, and I am on a number of committees with the Public Health Agency, the WHO, and CIHR. In terms of the outline for today and the learning objectives, I've outlined 4 of them. Firstly, to outline the general process for the WHO guidance development process during the COVID-19 pandemic in general, but with a focus on infection prevention and control, which is an area of expertise for me. To discuss the evidence review process and its application to guidance, and then to discuss the updating process itself, and finally a process for addressing research gaps and research innovation and development in this emerging area. Just begin by just making a small plug for why guidance needs really solid evidence and the sorting of the message. It's very interesting as a phenomenon during the course of this pandemic. We've seen mainstream and social media and absolute total overdrive. It's the first major pandemic in the modern age where you've got instant social messaging. There are experts and pseudo-experts who come on the media and portray themselves as experts and provide opinions, and mainstream media can't seem to get enough of it. We have many papers that are published even before they're reviewed. Science may be correct, but oftentimes premature, incorrect conclusions are provided. Some of these articles that are on preprint servers or published commentaries in the news media never are subject to peer review and are never in fact published. Even in the top quality peer-reviewed journals, there are articles that are premature, overinterpreted, and incomplete. And in fact, there have been several articles now that are outlining the very poor quality of a number of the studies that have been done in the pandemic because of this perceived urgent need to publish anything and everything. We see many letters and short reports, and there seems to be immense pressure with the journals to be first and fast in the publishing. So very important to carefully assess the evidence and to use critical appraisal skills on all of this information. Just to remind you about COVID-19 and SARS-CoV-2, the term COVID-19 comes from coronavirus disease, and it is caused by the SARS coronavirus 2, the ancestral or wild-type strain, and now multiple variants that have emerged. Just before we delve into some of the depths of the information I'm going to be speaking to you about, just a few basic viral facts to remind you about. The etiologic agent is SARS-CoV-2. We know there's a family of viruses. The first was SARS-CoV-1 that was associated with the first SARS coronavirus infection in the early part of the first decade of this millennium. It's a single-stranded RNA virus, and it's named for its unique morphology where you've got these crowns which are the spike proteins. We know that it's a respiratory virus predominantly, but has a wide repertoire of symptomatology associated with it. And interestingly, there's only a few coronaviruses that cause human illness. Most of them are respiratory. We've had a number of human coronaviruses. They are responsible for 1/3 of the common cold, but the more serious respiratory infections are SARS and MERS. And they use a unique ACE2 receptor that's very ubiquitous in humans, particularly in the respiratory tract. And there's a very high affinity for this receptor for the SARS virus, and particularly for SARS-CoV-2. And this is part of the explanation for its ultimate contagiousness, is that it may take only a few viruses required to cause infection. They're still working on this. We've recently completed some work and submitted on a preprint, just going into a journal, showing that in a hamster model, as few as 14 plaque-forming units, which is a very, very low number of viruses, are able to create invasive infection. So if this is true and parallels what we've seen in the human coronaviruses, very few infectious particles would be able to create infection. It's natural history, just to review very quickly, many of you will know this. The incubation period is 2 to 14 days with a median of about 5 days. Almost 98% will develop symptoms within 11 days if they're going to. And of those that are diagnosed, 80% will have self-limited, mainly respiratory illness. 15% go on to more severe illness and may require medical care or hospitalization. And about 5% go on to intensive care and or sometimes requiring ventilation. And this is the more severe part of the illness. After the initial viral phase, one moves into the fibroproliferative phase of the illness, where we start seeing ARDS develop, and of those that go on, anywhere from, I would say, 50 to 70%, up to 70% of those that are ventilated will have a mortal event as a result of this illness. We've now seen variants emerge. We knew that this was going to happen because we know the molecular clock for coronaviruses is about 2 mutations per month. We've known that for many years, so this was highly predictable. And there are changes in the virulence with the alpha strain, about 30 to 40% more transmissible, all the way up to about 70 to 90% for the gamma and delta variants, respectively. And carried with this is approximately 1.5 to 2-fold, some say 1.5 to 1.8-fold increase in hospitalizations. And the highest seems to be with the recent emergence of the delta. And just to remind you about the transmission routes, it's believed that this virus exploits multiple methods. We've got an infected individual, we've got large coarse droplets, we've got fine aerosols that may be spread in air-dependent mechanisms. We've got direct contact and then indirect contact via fomites, and then a susceptible individual. Just to remind you of the chain of transmission, we've got exposure, transmission, invasive infection. So you need exposure to these infectious microorganisms, in this case, infectious virus. Not all exposures lead to transmission and invasive infection, and we know that because there are families where some will develop SARS-CoV-2 infection, others will not. We know that exposure occurs when you're in contact with the infected source or the contaminated environment, and a whole series of events is required to create invasive infection: host susceptibility, presence of host receptors, receptivity of those host receptors, and we know that it's almost like crazy glue. I liken it to the ability of the SARS-CoV and its spike proteins to be able to latch on to those ACE2 receptors. We have issues with inoculum, viability, virulence, and then the effectiveness of hierarchy of controls. And as I mentioned, multiple routes are exploited by this virus. There's great debates about droplet and airborne. Suffice it to say that close contact seems to be the predominant route, whether that's through large droplets or smaller aerosol-borne There are settings in which longer airborne may occur, but it's very much dependent on the situation. We hear about restaurants and choirs and other things, but they're the exception rather than the rule. And it's important to recognize there's a continuum of droplet and airborne routes. Particle size becomes very important, and how these are generated in the human airway through coughing, sneezing, which produce large amounts of particles, and then talking, medical procedures. And then the amount of these particles and their distance that they're propelled is quite complex and dependent on a whole series of factors that are listed below. I'm not going to go through them all. And not all particles will contain infectious agents. And so that's very important to recognize when one sees papers that, oh, so many copies of RNA are expelled with a human breath if they're infected with SARS-CoV-2. Small numbers of copies does not equate to infectious virus, and that's very important to recognize. And then there are many factors with humidity, evaporation, directional airflow that become very important in how transmission occurs, and there's still a great deal of science to be done and data to come in to be able to develop a true sense of what are the variables associated with transmission with this virus. And just to show you where the SARS-CoV fits in terms of its reproductive number, it fits in very much with influenza, with the common cold viruses, in terms of its reproductive number, varying anywhere from 2.2 up to 2.68. And it's thought that the Delta variant may extend this by 40 up to approximately 80% for alpha and beta respectively. So it may extend it a little bit forward, and the fatality rates are anywhere in the 2 to 3% range, depending on the populations that are looked at. Just to turn our attention now to the focus of the development of guidance. This is just from the WHO website, and you can see when you open up coronavirus disease, you've got technical guidance, and all of the technical guidance is displayed there. And then you've got the research section, and the research section talks about the research and development blueprint, which in turn helps to guide and provide some of the evidence for the technical guidance. If we look at the technical guidance by topic, you can see that there's clinical preparedness, readiness, there's surveillance and rapid response, laboratory-based clinical care, serology testing, guidance for schools, workplaces, and then infection prevention and control. And then if you start pulling down those menus, you can see the many guidance documents that have been developed. Many of the national healthcare agencies, including the Public Health Agency of Canada, use these documents to help move forward the recommendations that are done on a countrywide basis. They may differ from country to country, but this is a core set of recommendations that are provided to all member states across the world, and including high, middle, and low-income countries. There are some differences in the interpretation and then the implementation, but nonetheless, this is a core set of guidances that are very highly evidence-based that are put together. With respect to the guidance development groups, there are a number of them. I'm going to focus on the infection prevention and control, of which I'm a member, along with close to 35 individuals from around the world who are expert in this area. And it's within the Health Emergencies Program, with experts within Preparedness, Readiness and Response to COVID-19. And these individuals are members of the Global Infection Prevention Control Network. They're recognized in their field for their expertise. They are often members of professional IPC organizations. Also, they have individuals who have experience in managing IPC outbreaks and have had previous experience with coronavirus or other respiratory viruses. And it also includes focal points from ministries of health. And there are designated roles and responsibilities. It isn't unpaid, it's a volunteer functionality for each individual that is coming forward to be a member of the GDG. The objectives of this GDG are to evaluate and critically appraise and interpret the available evidence, looking at benefits and harms. And if there is no specific agreement, then pre-published evidence and other evidence comes in that's pertinent, and it's summarized and presented. There's technical discussions and consensus building, as I mentioned, and if following all those deliberations, one is not able to achieve consensus, there can be a voting process in which individuals look at that, and I'll touch on how that looks later, but it looks at all aspects, including the evidence, the quality of that evidence, resource implications, values, preferences, ethics, and research gaps. The methodology for developing these guidelines is based on evidence and looking at the highest quality evidence, often if we can get them, randomized controlled trials or cluster randomized trials, rapid reviews, and then looking at evidence-based prior documents and ongoing evaluations of the available scientific evidence. It's a very robust and transparent process, and as I mentioned, looking at the evidence and the harms, the feasibilities, values, preferences, resource implications is very important, equity and ethics. And as I mentioned, when consensus is not reached, there's a survey that's done. It's somewhat akin to a voting process, but it allows one to be able to see what a temperature check is of the membership. And it's important to recognize that all opinions are brought forward. There's a rigorous discussion on the evidence and the interpretation of it. And then there's a fairly rigorous process following the development of a draft guideline. It's reviewed by an external review panel of experts, the World Health Emergencies Program Ad Hoc Experts Advisory Panel, and also the WHO Secretariat. And when this is being brought together, I just put this forward, as I mentioned to you, that the recommendation comes with looking at the strength of the recommendation and the level of evidence and the quality of that evidence, and then the balance of desirable and undesirable outcomes, the quality of the evidence, as I mentioned, values and preferences, resources, feasibility, acceptability, and equity. These are put into evidence to decision tables, and they're looked at and debated, brought forward, and as I mentioned, generally trying to achieve consensus, and if not, then a survey is done. Here's some of the beginnings of some of the existing WHO guidances. There was a document that was published after a very rigorous process in 2014. You can see on the left side of your screen. First brought out in 2013, but I think published in 2014. And this is infection prevention control for epidemic and pandemic prone acute respiratory infections. I was very engaged in this process to be able to put together this document from an editing process, and it laid some of the foundations in the preparation for pandemic prone acute respiratory, knowing that we might see another SARS or MERS at the time. And in fact, you can see the MERS guidelines that are present on the right side of your screen that have been developed. There are several principles that were elucidated and brought forward within the IPC strategies for suspected COVID-19, and this was based on this pandemic response document. Firstly is to ensure triage, early recognition, and source control. Secondly, applying standard precautions for all patients. Thirdly, implementing empiric additional precautions, so droplet and contact, airborne when necessary, implementing administrative controls, and then finally, environmental and engineering controls. With respect to triage, early recognition, and source, healthcare workers are encouraged to have a high level of clinical suspicion, do a point of care risk assessment, If in doubt, then use precautions. That's what we generally teach. Have a well-equipped triage station at the entrance to your facility, the entrance to your ward or to the room. You've got trained staff, and training is very important to ensure that they know how to uh, triage these individuals, instituting screening questionnaires, and you've seen those used throughout the pandemic. And there are case definitions, actually the ones that we've used, designed by the Public Health Agency of Canada and adopted and adapted in the provinces, have been extremely useful. Posting signs to alert healthcare workers and reminding symptomatic patients that they should come forward and present themselves. Applying standard precautions, so this is to ensure that there's respiratory etiquette, use of tissues when coughing, sneezing, offering medical masks to patients with suspected COVID-19. That's been employed throughout the pandemic with the thought that you're going to capture individuals who are coughing and sneezing. They've got virus-laden particles. Hand hygiene, very important, because people are coughing on their hands and then touching, shaking someone's hand, you've got the ability to transfer virus. Necessary to look at both alcohol-based hand rubs and soap and water. And if not visibly soiled, hand rubs are very convenient. If there's visible soil, then soap and water can be used in that. Implementing empiric additional precautions, contact and droplet precautions. This is your standard donning of gloves, gowns, medical masks, eye protection, be it goggles or face shields. You've seen that used throughout the pandemic. This requires education for your healthcare worker populations, and in my experience, nursing staff are well-prepared physicians. This was new for many of them, that, we've got to pay a lot of attention to the donning and doffing of the material when we're going in to see these patients in isolation. Very important, and it took a while, but everyone is familiar now with this and the importance, particularly errors in the doffing, because you've been spending a significant time in a patient's room, they're coughing, sneezing. Errors in doffing is a very important one. We learned lessons through SARS-1 about that, and we still see settings in which we see deficits in the doffing, which is very critically important. And then finally, disinfection of high-touch surfaces, airborne precautions for aerosol generating procedures, a well-ventilated room, and using an N95 or EU equivalent, the FFP2, in that setting for procedures such as intubation, or if there is CPAP, BiPAP, or other potential aerosol generating procedures. And a lot of debate about various professional societies considering all of their procedures aerosol generating, and generally those have to be worked through on a case-by-case basis. Administrative measures, this is a provision of adequate training, can't emphasize that enough. Very important to ensure your healthcare workers all the way down the line, from physicians, nurses, nursing aides, LPNs, pharmacy, nutritional services, cleaning staff, all of the members of the healthcare team need to be aware of the need for adequate training. Ensuring there's an adequate patient to staff ratio, which has been trying at times in the pandemic and the waves that have occurred, because we had healthcare workers who are becoming infected. Our experience in Alberta, the vast majority were picked up in the community. Establishing a surveillance process for acute respiratory infections, ensuring that everyone understands the process, and then if there's any illness, healthcare workers need to seek medical care promptly. We've had misses, as I'm sure everyone has, where healthcare workers have come to work ill, thinking it was allergies or forest fire smoke. We've seen this summer, and then working while symptomatic. And then, of course, compliance monitoring, and there are many coaching programs and audits that can be done, both healthcare worker aware and healthcare worker unaware audits, which the latter of which we've used and have been helpful. Environmental and engineering controls. This is to look at basic infrastructure in your healthcare facility, ensuring you've got good ventilation. Most hospitals have to meet Canadian Standards Association guidelines, so it's generally a given, but must be looked at. The importance of separation of at least 1 meter. We generally using 2 meters in most Canadian settings. ensuring cleaning and disinfection procedures are followed, particularly of high-touch surfaces. Managing laundry, food services, utensils, and a very important part of the process as well, and having here's some of the guidance documents that have been developed, and we'll just run through these from 2020 forward. There was the initial document when it was still called novel coronavirus, and then moving forward, that was back in January of 2020, then moving to IPC guidelines after home care was developed, almost at the same time, to look at both of these settings for when novel coronavirus, now SARS-CoV-2 infection suspected. Then there was an update in March of 2020, and then a whole series of documents through 2020 expanding into long-term care guidance, rational use of PPE, which is very important, particularly in low and middle income countries where there were shortages. It was a very difficult time in the spring and the first wave in many sites around the world where they were scrambling to be able to have inventory. We were lucky in many of our Canadian provinces where there had been storage of these. In many of the low to middle income countries, they were trying to grab supplies from the countries that were able to make them, and there were true shortages occurring. Then there was another update that occurred in IPC and hospitalized patients, risk assessment and exposure for healthcare workers, and then data templates for the healthcare worker, data dictionary to help out for exposure assessments for tools that were used. Then we had guidelines and guidance on identification and management of the infected healthcare worker, safe management of dead bodies. That was a very controversial item. What do we do with bodies and how to manage them? Body bags, use of masks in the children was a very controversial one, knowing that children tend to be affected less severely, less often hospitalized, and have very few mortal events. It's extremely rare in children to have fatal events with SARS-CoV-2 for a number of reasons that can be discussed at another time. Home care document update, then water, sanitation, hygiene, and waste management, so-called WASH document. Then an update on the transmission of SARS-CoV-2. Then following through in 2020, again, an update on rational use of personal protective equipment, another update on mask use, and then technical specifications for PPE as well. You can see the common ones that are used on display in this document. And then in 2021, again, another July 2021 update on IPC precautions in the acute care setting, quarantining of contacts, an aid memoir for vaccination activities. And this covered all settings from low to middle income countries up to high income countries, particularly when they didn't have adequate sanitation or in settings where there's no running water. And then this led to the adoption in Canada. This is probably very similar what we've done in Alberta to many other provinces. We have videos, posters. We have a COVID app that was developed through Spectrum, and I know that that's used in Saskatchewan as well. And particularly important in looking at the doffing, because we've seen many deficits in our healthcare worker unaware doffing audits where there are errors, and certainly you can pick up virus when you're doffing. We know that virus can sit on the masks, can be picked up on the gloves, and if there's sloppy technique, one may pick up the virus. So a critical juncture to ensure that each of the doffing steps, and that's where we found most of the errors, is in the doffing procedure. In terms of the research and development, there is another group that's been engaged, and that's the Infection Prevention and Control Research and Development Expert Group. I am the chair of this group and have been so since February of 2020. Its primary aim is to work in concert with the WHO Secretary and other experts with the purpose of facilitating coordination and collaboration of research and innovation in the field of IPC during the pandemic. And there are a number of objectives of this research and development expert group, and I'm not going to go through all of them. Supporting and engaging research in all aspects of IPC, continuing to review and update the guidance, enhancing timely sharing of IPC research results, contributing to the development and implementation of evidence-based recommendations. So hopefully the research and development is high quality evidence-based, enhancing global outbreak responses through evidence-based advice, dedicating special focus on research areas that are deliberated and decided upon and in consensus, contributing to research agendas in other areas that cross-fertilize IPC, such as in social sciences, behavioral sciences, environmental health sciences, and epidemiology, and particularly looking at modes of transmission. Understanding the impacts of COVID-19 with respect to antimicrobial resistance, very important because a lot of antimicrobials being used during the course of the superinfections, the bacterial superinfections that can occur after viral pneumonitis. And then finally, ensuring that the needs of low to middle income countries are looked at well. And here's the global research roadmap. This is a document available, and it's got the blueprint with 9 areas you can see there. This was formulated after a global meeting that was held in February in 2020, and in March this document came forward. I was engaged in helping with the production of aspects of this in the infection prevention control and including healthcare worker protection. But you can see this covered the gamut from viruses, the viral natural history, transmission dynamics, all the way through to ethics and social sciences. So it's looking at the totality of the evidence with respect to all of the aspects, and of course a big section on vaccines and therapeutics. And you can see here the principles guiding the implementation of this roadmap. We're powering research to control the outbreak in a coordinated manner, facilitating coordinated research to enable interventions, and a commitment to fair and equitable access to those at risk. You see this playing out now with high-income countries administering third doses when 2/3 of the world's population are having difficulty accessing a first dose. And this is a nice schematic because it actually shows many of those areas from the virus through to vaccines and therapeutics, clinical management, IPC, including healthcare worker protection, epidemiologic, and animal studies. One thing on the top upper right, you can see a large number of areas that cross-sect and intersect with IPC, including containment, control in healthcare community settings, home care settings, optimized PPE, and containment of contamination and aerosol spread. So a very hefty agenda in the IPC area with respect to this virus. And just to look at some of the areas that are knowledge gaps, and I'll touch on that a little later, modes and duration of transmission, still a major area of intense research. We've learned a lot, but there's still a great deal to be learned. Environmental stability, we've learned a lot, but more to be learned in environmental stability and proteinaceous secretions and other settings. There's some recent evidence to suggest a cocooning effect with the virus when it's in saliva or respiratory secretions as opposed to naked virus, which is used in the laboratory. Now to interpret that evidence in the setting of transmission from fomites and hands. Personal protective equipment. It's a large Canadian-led randomized controlled trial, which we're involved with in our center, and it's about 3/4 enrolled now. It'll be very important because it's a very high-quality study looking at a trial between the use of N95 respirators and regular medical masks in the setting of routine care. Interestingly, at the 50% enrollment, an independent safety monitoring board looked at this and saw no safety signal whatsoever in this very, very important Canadian-led study. The primary objectives of the WHO-IPC research group: understanding the effectiveness of public health and social measures, of which there are many that are listed there, and the IPC strategies to prevent secondary transmission in the community and healthcare settings, optimizing the effectiveness of PPE, and then minimizing the role of the environment in transmission. And here's some of the research and innovation achievements. They were provided in an April 2021 update, and you can see that Canada and the United States, North America, is very prominent in a lot of research work that's being done in respect to many aspects of SARS-CoV-2. There was a document that was published in April of 2021 with some of the achievements in IPC, and you can see on your right there are a number of studies that have been conducted where there's participation with University of Calgary. Also, you see University of Alberta, Oxford, where there's been a series of systematic reviews. There's studies on methods to detect cultivatable virus. That's one that I've been heavily engaged in. That is available in preprint and just submitted to publication. We've also got a project, one of my favorites, which I'll touch on, utilizing remotely piloted aerial systems, aka drones, to deliver medical supplies, PPE, and test kits to remote Indigenous communities in Alberta, and that study is on. There are also others you can see here that we were engaged in. There are many studies globally. There's probably 40 to 50. I'll just highlight some of the ones with which we've been engaged. A rapid conversion of a surgical orthopedic inpatient unit to COVID-19. We've published a couple of papers on this now. That was done in the foothills, where we used this ward because it was the largest with the most space and the best capacity, as opposed to an older part of the Foothills Hospital. Also work being done by the International Labour Organization, the WHO, on global surveys of healthcare workers. There's also an Infection Control Africa ICANN global survey on perceptions of healthcare workers that's being done, led by ISAC ICANN, and then also WHO headquarters looking at a study of international case control study. We are one of the only sites in Canada participating in Calgary in this healthcare worker case control study, which we hope to see completed later this year, to see what are the true risk factors for transmission to healthcare workers in the healthcare setting, not those who pick it up in the community, which we've seen quite a number of. And here's a photo to show you our drone. We've combined resources with Southern Alberta Institute of Technology, which has Western Canada's largest drone laboratory, where we've been successfully sending medical supplies and test kits using a Swiss drone, which is a helicopter style with a 50-kilo payload. And we've been able to show that we can send out test kits and bring them back to the laboratory without any degradation of signal. So that's been an important achievement, and we've now teamed with a trauma surgeon to show we can deliver remote ultrasound, which can be done over the web and or satellite, to be able to look for COVID lung in patients in remote settings. And I'll finally close now with some references, and if there's any questions and there's some time, I'll be pleased to be able to carry those questions. And I would like to thank you for your attention today during this presentation. Thanks very much.